Swayam Prabha. Digital India. Educated India.
This is Rangarajan here. We are going to continue the fourth lecture in the course on Eukaryotic Gene Expression. What we did in the previous three lectures is to briefly describe about eukaryotic RNA polymerases. We discussed about the various components of eukaryotic RNA polymerases, polymerase 1, 2 and 3, how they function and in the activation of gene expression in eukaryotes. Then we confined ourselves to understanding the core promoter elements and how variations within this core promoter elements can actually lead to differential gene regulation. Then I also very briefly mentioned about the various general transcription factor which interact with RNA polymerase and constitute what is called as a pre-initiation complex, and how variations within the general transcription factors itself, like their tissue specific expression and variations in recognition of core promoter elements, that itself can contribute to differential gene regulation. Now, what we are going to do today is to move a little bit away from the core promoter and then focus more towards proximal and distal promoter elements. And by the end of this class you will realize that the promoter actually consists of several kilobases of sequences upstream of the transcription start site, and a number of cis acting elements. You can call them as proximal promoter elements or distal promoter elements or enhancers. All these things together actually play a very important role in the regulation of gene expression in eukaryotes. So, we will also now study a little bit about the identification and characterization of various transcriptional activators which actually go and bind to these kind of a proximal and distal promoter elements, and binding of these transcriptional activators actually play a very important role in the modulation of levels of gene activity. And also the title of this course is Eukaryotic Gene Expression Basics and Benefits. So far I have not talked anything about the benefits arising out of eukaryotic gene regulation. Today maybe we will just spend about maybe 5 minutes to tell you how understanding the various promoter elements and transcription factors actually paved way for development of what are called as recombinant protein expression systems, by which you can actually express a number of recombinant proteins, and this has now become a big $1 billion biotechnology industry. So, the way that I have structured the course is that we will talk mostly about basics, but at the end of maybe about 4 or 5 lectures we will review the whole thing and really see, having studied the knowledge in the basic research, has it really led to any applications or any uses or any benefits have come out of some of this basic research. That is the way we are going to proceed further. So, let us now focus our attention on promoter elements which are kind of further away from the core promoter. So far we have been confining ourselves only to the core promoter region of a eukaryotic gene. Now, we all know the transcription starts from the transcription start site, and in all my lectures I indicate this as with an arrow. So, this is what is called as the +1. Now, what has been shown as people started studying about the various cis acting elements and various promoter sequences, it became very clear that in addition to the core promoter region, there is also a huge region. Sometimes this region, which is upstream region from the transcription start site, can run to several kilobases upstream, and if you actually make some deletions within this promoter region also, the expression of the downstream gene is modulated. You can see this is what I have written as a promoter activity, and for example let us now take this promoter region, which in addition to core promoter consists of further upstream sequences, and let us designate it the activity of this promoter, that is this promoter activity as let us say 6 pluses. That means it is very highly expressed. Now, people have started asking the question, in addition to the core promoter region, are there other promoter elements which are essential for the activity of the promoter? The way they went about is that you actually start doing deletion mapping, wherein you start deleting different regions of the upstream region and ask the question, how does these deletions in the further upstream region of the promoter affect the promoter activity? And this particular example you can see, when you make a deletion from here, when you delete this particular region, there is not much change in the promoter activity, suggesting that there may not be very important regulatory elements within this region of the promoter. But you can see when you delete further, when you delete from here to here, there is a drop in the promoter activity, and similarly when you make a deletion from here to here, there is a drop in the promoter activity, and so on and so forth. So this is what I illustrated here. So what this means is that when you make a deletion in this upstream region, there is a decrease in the promoter activity, suggesting that this region, which I inject in an orange box, may actually harbor some very important cis acting elements to which certain protein factors may be binding, and they in turn may be interacting with the general transcription factors and RNA polymerase in the chromotor protein and enhance the rate of initiation of transcription. Similarly, you can see there may be, in this particular case, there may be two other regions. Totally there are about 3 regions, and if you delete these promoter regions, there can be a decrease in the promoter activity, suggesting that this region of these promoters may harbor some important regulatory elements to which certain protein factors may be binding, and they in turn may be interacting with the core promoter region and contributing to transcription initiation. So it became very clear when you start studying about eukaryotic gene regulation. So far we have been talking only about basal transcription, where RNA polymerase 2 and general transcription factors are binding to the core promoter region, but it is now becoming very clear, in addition to this region, there are also other upstream regions which are essential for transcription activation of eukaryotic genes, and therefore these regions which are further upstream of the core promoters, to which certain transcription activators may be binding and further activating transcription, is refers to what is called as an activator dependent transcription. So, you have co-transcription or basal transcription contributed general transcription factor and RNA polymerase 2, and you have further upstream elements to which other transcription activators may be binding, and this is often referred to as the activator dependent transcription. Now, we are now going to spend maybe about 20 or 30 minutes, because so far I have been talking mostly about theoretical aspects. We also, it is very important for us also to understand how do people study the promoter activity, how do assay, what kind of experiments people normally do to assay promoters. What kind of experiments you actually do. So, what we are going to study in the next few minutes is to understand how does one assay the promoter activity. Now, how do you show that when you have this promoter region, the promoter is highly active, when you start making deletions, the promoter activity goes down. What kind of promoter activity assays people use to study actually the regulation of gene expression, and how does actually one identify the upstream regulatory elements. Okay, these are the 2 important techniques which have made a very important difference in eukaryotic gene regulation. Therefore, we will spend some time to understand these experimental techniques. Now, the promoter activity, how do you assay the promoter activity? One of the very important experimental systems that people have developed to assay promoter activity is by what are called as reporter gene assay in cell lines. Now, as you know, there are a number of eukaryotic cell lines which have become available, like HeLa cells, which are derived actually from a mammary tumor cell line, and you have liver cell lines, you have fibroblast cell lines, and so on and so forth. So, once you have these cell lines can be indefinitely cultured in the laboratory in vitro in a carbon dioxide incubator. So, you can actually use these cell lines, and you want to study a particular eukaryotic promoter. What you do is, for example, in the previous promoter, we know that there are probably 3 promoter regions which have been identified, and how do people identify these 3 promoter regions? What you actually do is, you take this entire promoter region, and downstream of the transcription sorted, you link them to a reporter gene. This is using recombinant DNA technologies. You can actually have restriction sites. By using restriction sites, you can actually put different reporter genes. Now, what is a reporter gene? A reporter gene is one which codes for a protein or an enzyme whose activity can be easily assayed. Okay, or whose presence can be easily assayed if it is a protein, or if it is an enzyme whose activity can be easily measured. So, it becomes very easy to assay the promoter activity. The most common reporter genes which are used in eukaryotic gene expression studies is the one that codes for a what is called as a green fluorescent protein. And you all know the people who actually discovered this green fluorescent protein, they actually won Nobel Prize a couple of years ago. And then there is something called beta galactosidase, which when you add a substrate, it actually, when the enzyme cleaves and you get a blue color, then you have what is called as a luciferase, which actually can convert luciferin in the presence of ATP, can act on luciferin and then emit light. So, the light can be very easily measured. Or you have reporter genes like chloramphenicol acetyl transferase, which converts chloramphenicol into acetyl chloramphenicol, and these non-acetylated forms of chloramphenicol can be easily separated on a thin layer chromatography, and the number of acetyl, the percent of acetylated form that is formed is actually a measure of the promoter activity. So, these are the most commonly used reporter genes in eukaryotic gene expression studies. So, when you want to study a particular promoter, for example, in this particular case, let us say you are interested in here, you just clone a reporter gene downstream of this promoter, and this entire thing is actually put in a plasmid, and this is called as a reporter plasmid, where you have a promoter of your interest and you have a downstream reporter gene, and you then transfect this into various cell lines. Okay, and there are various methods for transfecting gene plasmids or genes into cell lines. You can do what is called as a calcium phosphate precipitation, you can do what is called as a lipofection and so on. There are a number of techniques, but sufficient to know that you can actually introduce or exogenous DNA into eukaryotic mammalian cell lines in culture, and this plasmid DNA goes inside, and the various protein factors which are present inside the nuclei of these cells actually will now go and bind to this promoter and activate the reporter gene expression, and then you actually harvest these cells, lyse the cells, and say for reporter activity. Okay. So, this is how in the previous slide I showed you that when you start cloning different deletion regions of the promoter region downstream of reporter gene, the reporter gene activity started varying, and these kinds of studies actually told in that case that there are actually 3 promoter regions which when you make deletions the reporter gene expression actually goes down. So, the assaying the promoters using reporter genes in various cell lines made a very important contribution for understanding the promoters of a number of eukaryotic genes. The other important promoter activity study that actually made a very important contribution for understanding eukaryotic gene regulation is using what is called as cell free transcription studies. So, here what you do is, suppose you want to study a gene which is present in a liver cell line or say HeLa cells. So, you actually make whole cell extracts or nuclear extracts from these cells. Okay. And this preparation of nuclear extracts which are transcriptionally competent requires a lot of required a lot of effort and lot of studies have gone into it. Just like you cannot make, there is a very standard procedure for actually making nuclear extracts which are capable of transcribing eukaryotic promoters in vitro. So, making transcription competent nuclear extracts or whole cell extracts from mammalian cell lines has made a very important contribution for understanding eukaryotic gene expression. So, once you have what is called as a cell free extract, okay, or a soluble extract which contains all the factors including RNA polymerase II, general transcription factors and your other upstream activators in that particular cell line, then what you do, you put a what is called as a DNA template for in vitro transcription. And this DNA template actually is a lot of people have been using all kinds of templates, but one very interesting and very innovative template which was designed by Savadogo and Roeder is what is called as a G-free cassette. Now, what you actually do, for example, if you are interested in studying this particular promoter which contains a data box and transcription start site here, a cell free transcription system or an in vitro transcription system, what you do is, just like we cloned a reporter gene downstream of the transcription start site for the transfection assays, in this particular case you actually clone what is called as a G-free cassette downstream of this promoter region. Now, what is a G-free cassette? A G-free cassette is a guanosine free region or a G-free region cloned immediately downstream of the promoter. That means this template actually contains only C, A and T residues, it has no G residues. So, this template can actually transcribe in the absence of GTP, because GTP will not be incorporated into the RNA. Therefore, this is a G-free template. So, when you actually carry out the transcription in presence of RNase T1 won't methyl GTP, the transcripts which are initiated from places other than transcription start site will be aborted, and only those transcripts which are originating from the transcription start site will be visualized, and by actually cloning G-free cassettes of different lengths you can actually get different sizes of RNA, because immediately after the G-free region you again have the normal DNA which contains G, and therefore the O-methyl GTP which is there in your reaction mixture will get incorporated and transcription will not proceed further. So, only your G-free template will be transcribed, and depending upon the size of your G-free cassette you will get that particular size RNA, because you are actually in all these assays you use alpha p32 UTP, radio label UTP as one of the nucleotides, and therefore the RNA gets radio labeled, and you can actually run this labeled RNA after the reaction is over and then analyze them on a acrylamide gel, and depending upon the size of the G-free cassette you can get a very specific size product. For example, in this case the G-free cassette is of 360 base pairs in length, and as you can see, if you can actually use only a promoter which contains only the core promoter, you get a very low levels of transcript. But the moment you use a template which also contain upstream region, you can see there is a manifold increase in the level of RNA that is actually made in the cell-free system, clearly indicating that this region of the promoter actually harbors very important cis-acting elements to which certain transcription factors are going and binding, and as a result of it the transcription is enhanced several fold. So, the difference between this and this this and this is actually is contributed by the transcriptional activators. So, this is basal transcription, this is the activator dependent transcription that you are measuring. So, by using transfection assays in vivo in cell lines or by using cell-free transcription studies, one can actually assay the activity of various promoters. The number of such cell-free transcription systems have been developed over the years. Some of the most popular examples are, for example, you can actually make nuclear extract from HeLa cells. As I told you, these are cells derived from a mammary tumor. You can actually make such kind of a transcription competent cell-free extract from Drosophila embryos. You can actually make from liver, rat liver, and so on and so forth. So, in all these cases, you actually can actually either make a crude nuclear extract which contains everything, but as research proceeded, especially in the late 80s and 90s, and once people started purifying various general transcription factor, instead of using crude nuclear extract, people actually started using what is called as a in vitro reconstituted system, wherein you have purified RNA polymerase and its subunit, and you actually have purified general transcription factors. You add this very specific components, and this is actually called as a recombinant or reconstituted in vitro transcription system, where instead of taking just crude nuclear extracts, you have purified components which contains RNA polymerase 2 and general transcription factor. You can put them together, and then you add your DNA template, nucleotides, and you can actually measure the rate of transcription. And as people started cloning the various genes for the general transcription factors, they have also been able to express the general transcription factors in E. coli systems, and people started using recombinant general transcription factor in these cell-free transcription systems. So, you have RNA polymerase 2 purified from a particular source, and then you add recombinant TF2D, recombinant TF2B, and so on and so forth. So, now you have a reconstituted system where you know exactly what you are adding. So, from crude nuclear extracts, purified components, and recombinant general transcription factors, we now have evolved using highly specified in vitro transcription systems. Okay. Now, in addition to this G-free cassette, which is one of the very popular methods of measuring in vitro transcription assays, you can also do what is called as a primer extension, where so the template here is slightly different from what I showed in the previous slide. Here you do not have a G-free cassette, but you have your normal gene sequence which is present, which is the coding region of the normal gene, and you do the transcription, and then once you have done the transcription, you do what is called as a primer extension. So, here the RNA is not labeled. You actually carry out a regular non-labeled RNA. You take this RNA, and then once the RNA is synthesized in the cell-free transcription system, you hybridize to a small primer, and then extend the primer using a reverse transcriptase. So, suppose the transcription starts here, the primer will be extended up to the transcription start site, and then this will be radio labeled, and this can now be analyzed on a gel, and you can see when you have no template, only the radio labeled nucleotide, only the other components of the thing, you do not get a primer extended product. But when you put the template and when you have all the components for transcription, this template, the transcription will start and RNA will be made, and you can get a specific extended product which is called as the primer extended product. So, either using a G-free transcription template or using what are called as primer extension, you can actually quantify the RNA that is synthesized in a cell-free transcription system, and by using promoters whether they contain mutations or deletions, you can assay all these promoters in these in vitro transcription systems, and you can actually demonstrate what kind of regions in the promoter are actually essential for activator dependent transcription. So, this is how people have actually identified various promoter regions which are essential for transcription activation of genes in eukaryotic cells. Now, okay, once you know that this particular promoter region is essential or plays a very important role in the activation of transcription, the next question comes, how do you identify what kind of transcription factors are actually going and binding to these promoter regions? As we know, if a particular promoter region, when you delete and thereby there is a decrease in promoter activity, it actually means that there are certain protein factors which are transcription factors which are actually going and binding to those regions, and that is how this promoter region became very important. So, if you delete the promoter region, these transcription factors can no longer bind to those regions, and therefore there is a decrease in the promoter activity. So, once you identify these kinds of promoter regions which are important for the promoter activity, the next question is, how do you actually identify what kind of protein factors are actually going and bind to these promoter regions. Now there are many assays, many experimental techniques by which you can actually identify these transcription factors binding sites in the promoter as well as the transcription factors themselves. So let us now study one or two techniques which people routinely use to identify transcription factor binding sites in the promoter regions. One of the very popular assays that people use for identifying the various promoter regions which are essential for binding to protein factors is called as electrophoretic mobility shift assay. It is very popular known as EMSA. EMSA, electrophoretic mobility shift assay. Now let us understand what this EMSA actually is. Now for example, I told you in my previous study I had a promoter, and when I had deleted this particular region of the promoter, there was a decrease in the promoter activity, right. Now I want to really see, does this promoter region really bind to any transcription factor? And it is because of this transcription factor binding that this promoter region actually contributed to promoter activity. How do I go about it? What you do is that, let us say this is about say 50 or 60 base pair region, which when I deleted, the promoter activity dropped down. So what I do is I make a double stranded oligonucleotide. These days we can actually make oligonucleotides of varying length. You can in fact even synthesize genes these days without any problem. People have been making synthetic genomes these days. So making small DNA fragments is not a big deal. Therefore you actually make a double stranded DNA oligonucleotide, and then you radioactively label this oligonucleotide. So you have a region which is actually essential for promoter activity in vivo, and you take this DNA fragment, and as a control you also take an adjacent promoter DNA fragment, which when I deleted, it had no effect on the promoter activity. So I have an oligonucleotide which in the promoter context has an important role in the promoter function. I have an adjacent region which when deleted, nothing happened to promoter activity. So this serves as a control. Now what I do, I take a cell extract or I take a tissue extract in which this gene is normally active, and ask the question, what kind of protein factors within this tissue extract or cell extract is actually binding to this region, and as a result there is an increase in the promoter activity. So what I do is that I take this radio-labeled DNA fragment and then incubate it with a nuclear extract or a whole cell extract of my interest. From whatever tissue I am interested in, I make this whole cell extract, and once I incubate this radio-labeled DNA with this nuclear extract in presence of what are called as certain amounts of non-specific DNA, therefore the non-specific interactions are brought about or knocked off, and then you actually do what is called an electrophoresis. In a polyacrylamide gel, you do an electrophoresis. When you do an electrophoresis, what happens? When the DNA is not bound to any particular protein, it will move faster, and therefore it will have faster mobility. But if this DNA fragment is interacting with a specific protein inside these nuclear extracts and is forming a complex, and because of the formation of the DNA protein complex, the mobility of this DNA gets shifted. That is what is called an electrophoretic mobility shift assay. So the mobility of the DNA gets shifted, and therefore you have a retarded complex, and therefore these assays are even called as gel retardation assay, where the mobility of the DNA gets retarded because of the formation of a DNA protein complex. So in this particular case, this particular DNA fragment was able to form a DNA protein complex, and therefore the mobility of this DNA got retarded here or shifted here, whereas this particular promoter region, which did not have any role in the promoter function, did not bind to any particular protein in that particular cell extract, therefore it did not form any complex and it was moving much faster. So you can see, by using these kind of electrophoretic mobility shifts assays, you can actually identify what kind of protein factors are actually present in this region, in these particular cell extracts, and are they really binding to these promoter regions. And this assay is very popular and very easy to use, because you can also distinguish between specific versus non-specific binding. In this case, for example, I have radio labeled both the specific DNA as well as non-specific DNA, and I am showing that only the specific DNA is binding. There are other variations by which you can actually do is, you can also do, for example, you can, once you get this radio labeled DNA protein complex, you now add excess amounts, 10-fold, 20-fold, 50-fold or 100-fold excess of the unlabeled DNA of the same unlabeled DNA, and you can see now the complex is completely chased. Okay. Whereas when you add the same unlabeled excess component of the other non-specific DNA or the DNA which does not bind, it does not chase. So clearly these are called as competition experiments, which again tells you that you are actually looking at a very specific DNA protein interaction. Only a homologous DNA fragment is able to chase the binding of the protein to the radio labeled DNA, whereas a heterologous DNA fragment cannot chase or cannot compete for the binding of the protein in the nuclear extract to the specific DNA. So, by using these kinds of competition experiments, you can actually convince yourself that what you are actually measuring is a very specific DNA protein interaction. The protein that is actually binding to this DNA fragment is very specific, but it does not bind anywhere else. Now, there is another very popular experimental technique that people have been using, and this is actually called as a DNase I footprinting. Okay. So, in addition to the EMSA, this DNase I footprinting also is a very popular and very important technique that people have been using to identify transcription factor binding sites within the eukaryotic promoters. Now, let us see how exactly it is done. Now, let us say for example, I have a promoter region and I suspect there is a protein actually that is binding to this particular region within this promoter. Now, when I take this promoter DNA and then I radio label it at one end of this DNA, remember this is very important. When you do DNase I footprinting, you have to radio label the DNA either at the 5' end or at this end. You cannot label both ends, you can only label one end of the DNA is radioactively labeled. Then you start treating it with very limited amounts of DNase I. As you know, DNase I introduces double stranded DNA nicks. When you have manganese and magnesium as cations in the buffer, it introduces double stranded DNA nicks and it will randomly cleave the DNA. In this particular case, for example, these are all the DNA. For example, the first instance DNase I will make a nick here, and as a result you will get a radio labeled DNA fragment of this size. When it makes a nick here, you will get a radio labeled fragment of this size, and when it makes a nick here, you will get a fragment of this size, and so on and so forth. So, depending upon where the DNase I is making a nick, you will get different sizes of the radio labeled fragment. But here the DNase I does not distinguish, it is a very random process. So, it has a equal probability of cutting the DNA at any particular region within the DNA. So, almost all the regions of the DNA are equally susceptible for DNase I attack. Okay. Now, let us see what happens. But suppose now you take this radioactive DNA and then incubate it with your protein of your interest, like what we did in the previous experiment. Suppose you now incubate with the nuclear extract, so that this particular a specific protein present in the DNA now goes and binds to this specific region within the promoter region. So, you now have a DNA protein complex formed here. Now, when you take this DNA template and then treat it with DNA, as you can see here, the DNase cannot, while it has cut here, it cannot cut this region because there is a transcription factor binding here, and because of steric hindrance it will not allow DNase I to act there. So, therefore, the DNase only can cleave here, here and here, but not in this region. So, therefore, it cannot generate these two radio labeled fragments here. So, when you take this mixture of fragments and this mixture of fragments and analyze them on a polyacrylamide gel, on a denaturing polyacrylamide gel, while you get uniformly radio labeled DNA fragments of all size when there is no protein binding, in this particular case you will not see DNA fragments corresponding to this region. So, this region actually now forms what is called as a footprint. So, the region where exactly the transcription factor bound actually leaves a footprint, and this is what is called as a DNase I footprinting studies. Okay. These are an actual gel looks from our own lab. We had done some of these footprinting studies, and when you do this, label this promoter and do this kind of DNase I footprinting in the absence of the binding protein, you see you have radio labeled fragment generated all through. But when you now incubate the same DNA with a specific protein, which I know binding a specific region, and you can see this particular region is not susceptible to DNase attack, and therefore I conclude this is the region where my transcription factor is actually binding. Okay. And these are the actual gels. This is from literature where people have actually done. You can see in the absence of protein you have radio labeled fragments of all sizes. When you have a transcription factor and when it is binding in this region, DNase I cannot chew this region, and therefore you do not see band in this region. Now, our own lab have actually done some of these DNase I footprinting studies. I will discuss these experiments in much more detail in the next class when I talk about transcription activators and so on and so forth. But suffice for it to know here, for example, in this case we have taken a promoter region of a gene called alcohol oxidase in a yeast called Pichia pastores, and we are interested in finding out what kind of transcription factor is actually binding to the promoter region of this particular gene. So, we radioactively label this promoter region and did this DNase I footprinting study, and you can see here now I have got a footprint here. So, the first two, these two lanes are without protein and these 4 lanes are with protein, while all other regions are getting cleaved. You can see this region is not susceptible to DNase I. So, I conclude that this is where the protein is binding. Now, I can also label the other end of the DNA and repeat the same thing. So, I will get the protein binding region both in the top strand as well as the bottom strand. So, this is the footprint in the top strand of the DNA, this is the footprint in the bottom strand of the DNA. By putting this together, I can actually tell you this is exactly the region where the protein is actually binding. The protein is making contact both in the top strand as well as in the bottom strand, and therefore it is binding like that. So this is how one can precisely map transcription factor binding sites in the promoter region using DNase I footprinting studies. So using assays such as EMSA, DNase I footprinting, as well as transfection of reporter plasmids into various cell lines, making promoter deletion studies, as well as cell-free transcription studies, either using crude nuclear extracts or using purified components of general transcription factor, people have been able to and using these assays, people have been able to purify a number of transcription factors from various eukaryotic cells, and have also been able to identify the various promoter elements to which these transcription factors are going to bind. Okay. So once you have purified these proteins by biochemical means, then you can actually use these purified proteins, and then you can, by raising antibodies against it and by using routine recombinant DNA and genetic engineering techniques, you can actually clone the genes. So over about between actually 1980s and 2000, almost 2 decades, a number of transcription factor genes were actually cloned. So genes encoding various transcription factors were actually cloned. So we have actually the genes which are actually coding for these transcription factors, and we have also gained the knowledge where exactly in a promoter region of a particular gene these protein factors are actually binding. So once you have the gene coding for these eukaryotic transcription factors, what you now do is, using recombinant DNA technology, you can actually express these recombinant proteins in a E. coli system. So you have a eukaryotic transcription factor which has been produced in large amounts in E. coli cells. Here I have shown, for example, we have actually overexpressed one of the transcription factors which we are interested. We actually cloned the gene coding for this transcription factor, we have overexpressed this protein in E. coli cells, and we actually purified this recombinant protein from E cells. This is what is called a Kumasi blue stain SDS polyacrylamide gel electrophoresis. This is the purified transcription factor, and now actually I can take this transcription factor and put them in my in vitro transcription system or a cell-free transcription system, and actually show that when I have the transcription, in the absence of the transcription factor, I do not see very high levels of transcription, but the moment I put the transcription factor, that goes and binds to this transcription binding site in the promoter region, and therefore I get very high levels of the transcript, clearly indicating that. So this is the kind of so I first took the promoter region, made some deletions or mutations, and identified what are the regions essential for promoter activity. Then by using EMSA DNase I footprinting as an assay system, I went ahead and purified the transcription factor which actually binding to this region. Then I have actually cloned the actual gene that code for the transcription factor, and then I have taken the gene, put this gene in E. coli system, made the recombinant transcription factor. Now I put this recombinant transcription factor back into my cell-free system and convincingly show, yes, this recombinant transcription factor can in fact activate transcription from that particular promoter region. So the circle is complete. So this is how people have functionally demonstrated that the genes that they have cloned or they have identified actually encode transcription factors, and functionally they can demonstrate that these genes can actually function as the recombinant proteins can function as transcription factors in vitro systems. You can also assay the transcription factor function of these cloned genes in a in vivo system, and a very popular assay which has again made a very important contribution to understanding gene regulation is called as cis-trans co-transfection assay. What did you see in the previous experiment? I took the recombinant protein made in E. coli cells, put it in a cell-free system, and showed that this transcription factor can activate transcription from that particular promoter region. But in this case, I can also argue that you have only shown in a in vitro system. You have actually not showed in a in vivo this gene actually codes for a transcription factor. So for that, what I do is I make two plasmids. One is called as a reporter plasmid, another is called as an expression plasmid. So I take the gene which is actually coding for a particular transcription factor and clone it in front of what is called as a constitutive promoter. That is, this is a promoter which is expressed all the time. Remember, in our eukaryotic cells we have promoters which are constitutively expressed, and there are promoters which are inducible, because we do not have to make all the proteins all the time. We only make proteins all the time, probably those involved in glycolysis, Krebs cycle, fatty acid metabolism, protein metabolism. These are all, every cell in the body requires these enzymes and proteins, and therefore many of the gene coding for these enzymes and proteins will be constitutively expressed. So these are called as housekeeping genes. despite what irrespective of what cell type you are dealing with, almost all the cells actually express this set of enzymes. So these are called housekeeping functions. Okay, so you need this protein, so all the cells will make these proteins, but there are many proteins which are actually expressed in specific cell types and at specific periods. That is, these genes are expressed only when there is a need for them. So these are called as inducible genes. Okay. So by studying a number of eukaryotic promoters and eukaryotic genes, over the years people have identified a number of inducible promoters, number of constitutive promoters. So you take the gene coding for your particular transcription factor, you clone it in front of a constitutive promoter, and you also take the promoter region which you suspect is the binding site for this particular transcription factor and clone that in front of a reporter gene, which we have discussed earlier. Now you mix both this plasmid and transfer them into eukaryotic cells of your choice. So that is why it's called, this is called a cis plasmid, this is called a trans plasmid. That's why this assay is called a cis trans co-transfection assay. So two plasmids are transfected simultaneously. Now what happens? This transcription factor gene will now be expressed because it has a constitutive promoter, and the transcription factor which is made from this particular plasmid will now go and bind to this particular binding site present in the promoter region, and then as a result it will activate this promoter, the reporter gene will be expressed. Now you can easily assay the reporter gene activity. So by making recombinant transcription factors and examining their transcription activation potential in cell-free systems, as well as by doing these kinds of studies in vivo, you can actually convincingly demonstrate, yes, what you really have is a gene that codes for a transcription factor which can activate transcription either in a cell-free system or in a in vivo system using cell lines. So genes encoding mammalian transcription factors were cloned into mammalian expression vectors, and these were transfected into mammalian cells, and their transactivation functions were studied by using these kinds of cis trans co-transfection assays. So genes encoding transcription factors, that's what I mentioned here, all these assays were done, and as a result of all these experiments, a number of upstream activation sequence enhancer elements have been identified in a number of eukaryotic promoters. Okay. So all that you have to do, suppose I want to study the particular promoter, I can make these deletions, put it in front of reporter gene, test them in cell lines, or I can also make these, put it in front of G-free cassette and then assay them in a cell-free system, and then I can also use techniques like EMSA and DNase footprinting and then really see what kind of protein factors actually binding to these promoter elements, and by doing this combination of studies you can actually identify exactly what kind of promoter regions are actually acting as enhancer sequences and what kind of transcription factors are actually going and binding to these sequences. So as a result, today we are actually flooded with hundreds and hundreds of eukaryotic promoters and eukaryotic gene sequences. Okay, and you can actually classify many of these promoters as strong promoters, weak promoters, constitutive promoters, inducible promoters, and so on, which I explained here. When you say a strong promoter, what it actually means is that probably it has multiple enhancer elements or multiple upstream elements to which a number of transcription factors are binding, and as a result they can synergistically interact with the general transcription machinery and therefore can recruit RNA polymerase II much more efficiently the promoter, and therefore the rate of initiation is very, very high. It can initiate multiple rounds of transcription activation initiation in a given time. So it's a strong promoter. In a weak promoter, probably a weak transcription factor is binding to it, or probably not many transcription factor binding sites are present in the promoter, and as a result they cannot efficiently recruit the general transcription machinery to the transcription initiation site, and therefore these promoters are transcribed rather weakly. And then I mentioned in the just previously that there are also what is called constitutive promoters and inducible promoters. In the case of inducible promoters, the transcription factor which go and bind to these enhancer elements, they bind only under certain conditions which you have to induce. Okay, we will discuss a little bit more in detail in the next few minutes. So it became very clear transcription activation in mammalian cells is a very, very complicated process. Okay, so it not only involves a core promoter region to which the general transcription factors and RNA polymerase II binds and then constitute what is called the basal transcription, but there are also many protein factors which are binding to many upstream elements in the promoter region, and it is the interactions between these transcription factors and amongst the general transcription machinery, these synergistic interactions ultimately contribute to the increase in levels of transcription of eukaryotic protein coding genes. So the structural and functionals, now we will go now one step further and see. So you had purified transcription factors from various sources, they have actually cloned the genes, we have overexpressed the proteins, and once they demonstrated that they have indeed cloned the gene coding for a transcription factor, or they have large amounts of this eukaryotic transcription factor in their hands, then people started looking at how do these transcription factors go and bind DNA, and how do they activate transcription either in a cell-free system or in a cell line transfection system. So then people by these kinds of studies, it became very clear that the transcription factors are actually modular proteins, and they actually are composed of distinct and separable functional domains. Okay. Now the two important domains actually emerged. Now remember, when you say we have a transcription factor, it has to go and bind to a specific sequence in the promoter, and once it binds, it had to interact with the general transcription machinery and activate transcription. So most of the transcription factors which people studied, they at least contained two major domains. One is called as a DNA binding domain for interaction with specific DNA sequences, and it also contains what is called transcription activation domain, which will now, once it binds DNA through this transcription activation domain, it interacts with general transcription factor and enhances the rate of transcription initiation. So almost all the eukaryotic transcription activators contain two major functional domains, a specific DNA binding domain and a specific transcription activation domain. So these are the two minimal domains or modules that a eukaryotic transcription factor must have. And we will study this a little bit later, but suffice for you to know that the function of both the DNA binding domain and the transcription activation domain of eukaryotic transcription factors can also be modulated by post translational modifications. And the next few classes I will elaborate on many of these examples. Post translation modifications of transcription factors is one of the major mechanism by which the transcription factor function is modulated in eukaryotic cells. For example, there may be a transcription factor present in the cell, okay, but now it may be present, it may be even be binding to DNA, but it will not be able to activate transcription. But if there is a kinase and this kinase goes and phosphorylates the transcription activation domain, now the transcription factor becomes transcriptionally active, and then it can interact with general transcription machinery and activate transcription. So in this case, phosphorylation of a specific amino acid residue in the transcription activation domain of that particular transcription factor is the mechanism by which the activity of that particular gene is modulated. So phosphorylation, which is a post translational modification, is a major regulatory mechanism in this particular example. And there may also be examples where the transcription factor actually may be present in the nucleus, in the cytoplasm, in the conditions when the gene is not active, and when the gene needs to be activated, a specific stimulus takes this transcription factor and translocate the protein to the nucleus so that it can actually bind to DNA and then activate transcription of certain genes. So there are a number of mechanisms, and in many cases post translational modifications such as phosphorylation, acetylation, ubiquitinylation, and so many other post translational modification of specific amino acids of the transcription factors play a very important role in regulating the activity of these various transcription factors. Now if you now today want to know what kind of eukaryotic transcription factors are actually present, how many transcription factors have been identified, what kind of sequences they go and bind, you simply go and then click on this website, and you have a whole bunch of information about the various eukaryotic transcription factors that have been identified so far and what kind of sequences they actually go and bind. So the number of transcription factors have been identified, their genes have been cloned, and people have even identified what kind of exact promoter sequences these transcription factors will go and bind. So this kind of information is now available. All that you have to do is to go to this particular website, and there are many such websites. This is just one example where you can get this kind of information. So what I will do in the next few minutes is to actually give you some key examples just for you to get a flavor. But remember, in the next few classes we are going to take specific examples of certain genes on certain transcription factors and study in detail, but for all that I have told you so far, for you to get an idea how actually transcription factors will activate, I am going to give you 2 or 3 examples. Okay. For example, there are genes called as heat shock genes, and these genes are active only under conditions of heat shock. Now as long as you grow these mammalian cells at 37 degrees centigrade, which is the normal temperature, body temperature, these genes are not active, these genes are not transcribed. But when you increase the temperature from 37 degrees to 42 degrees, this is what is called the heat shock, then certain genes called heat shock genes are activated. Now people have gone ahead and asked the question, how does this heat shock help the activation of transcription of these particular heat shock genes? What people have actually found out in such cases, using the various techniques that I have described, people have actually shown that there is a enhancer element in these promoters of these heat shock genes called as heat shock element. And all these genes which are activated by heat shock, if you go to the promoters, they contain these heat shock elements. And it turns out there is a transcription factor called heat shock transcription factor which actually can recognize this particular sequence. So when you give a heat shock, this transcription factor goes and binds to this particular heat shock element and then enhances the rate of transcription initiation of these genes. So all these genes which are induced in response to heat shock, you can see now there is a common mechanism. Whenever there is a heat shock, whenever there is an increase in temperature from 37 to 42, this heat shock transcription factor goes and binds the heat shock elements of these various promoters of these various heat shock genes, and all these genes are getting transcribed. Similarly, there are what are called as metallothionein gene. Okay. Now this gene need not be expressed in normal times. This gene will be expressed only when there is a metal concentration goes up inside the cells. So it turns out when you now look at the promoter regions of these kind of metallothionein genes, there is what is called as a binding site for a transcription factor called ACE1 in yeast cells. Okay. In this case we are talking about yeast cells, and it turns out when there is an increase in metal concentration left, for example copper sulfate, when you put copper sulfate or copper chloride in the culture medium of yeast cells, now this copper now goes and binds to this particular transcription factor, and as a result in presence of copper this ACE1 can bind to this particular sequence in the metallothionein promoter and enhance the rate of transcription initiation. So metallothionein genes are transcribed only when you have metals like copper in large amounts, because now this copper binds to this transcription factor and make it transcriptionally active, therefore it can go and then activate the transcription of these genes. The most versatile or the most well studied example of inducible gene expression in higher eukaryotes is what is called as the mechanism by which genes are actually activated by various steroid hormones. Now you know there are many steroid hormones. Steroid hormones are nothing but very small hydrophobic molecules. For example, you have glucocorticoids, you have progesterone, you have estrogen, androgens, mineralocorticoids. These are all called steroid hormones. Now all these steroid hormones function. You know the estrogen has a specific function during reproduction. Progesterone has a different function, glucocorticoids has a totally different function, but it turns out all these molecules, although they are structurally different and have different physiological function, the mechanism by which they act appears to be similar. It turns out all these molecules, which are all called as collectively called as steroid hormones, when you expose cells to these steroid hormones, being hydrophobic, these steroid hormones traverse the cell membrane and then they enter the cytoplasm, and inside the cytoplasm they have actually receptors which are present. For example, if you take glucocorticoids, there is a specific receptor called glucocorticoid receptor which is present in the cytoplasm of these eukaryotic cells. Now the glucocorticoid hormone, when you add, it goes and binds to this glucocorticoid receptor, and when there is no hormone, this glucocorticoid receptor is actually present in the cytoplasm. This is because in the absence of the hormone, this glucocorticoid receptor interacts with the protein called heat shock protein 90 HSP90, and as a result of this interaction this receptor cannot go inside the nucleus. Remember, if you want to activate the transcription of genes, the transcription factor has to go and bind to the promoter elements of the genes which are actually inside the nucleus. So when there is no hormone, the glucocorticoid receptor stays in the cytoplasm because it is complex with the protein called HSP90, and therefore it cannot activate the transcription of genes which are responsible for glucocorticoid hormone. Now the moment you add glucocorticoid hormone, being hydrophobic, the glucocorticoid hormone simply traverses the cell membrane. Now it goes and binds to the glucocorticoid receptor. Now when the hormone binds to the receptor, it induces a conformational change in the glucocorticoid receptor, and as a result the heat shock protein can no longer bind to the receptor. So the heat shock protein dissociates. So now we have a glucocorticoid receptor which is not in complex with heat shock 90. And it turns out when this kind of a conformational change, the hormone binding also exposes what is called as a nuclear localization signal. Now for many of the proteins to go inside the nucleus, they have to contain what is called as a nuclear localization signals. So only those proteins which have this nuclear localization signal or NLS can actually go into the nucleus. So as long as the glucocorticoid receptor in the absence of hormone is interacting with HSP90, this nuclear localization signal is buried inside the protein and is therefore not exposed. and that is the reason the protein cannot go inside the nucleus. But once the HSP90 dissociate, this nuclear localized signal now gets exposed, and as a result the receptor can now go inside the nucleus and bind to what are called as glucocorticoid response elements, which are specific binding sites for this particular receptor, which are present in all the genes which are responsive to glucocorticoids. And therefore, now the receptor binds to these specific elements and then activates the transcription of all these genes. So, you can see in one case the heat shock induced the transcription factor that went and bound to the promoter and activated genes. I give another example where in the presence of a metal, activity of a transcription factor is modulated. In the presence of metal, the protein is able to bind to DNA and therefore activate transcription. Here I have another small molecule which actually the regulation is at the level of nucleocytoplasmic transport of the transcription factor. When this small molecule is there, the transcription factor translocates from the cytoplasm to the nucleus, then binds to specific response elements or specific enhancer elements in the promoter regions and activates the transcription of the downstream genes. There is another very interesting example and a very important transcription factor called nuclear factor kappa B, which is a very important transcription factor involved in inflammatory response, and it belongs to a super family of transcription factor which is called as the REL family of transcription factors. Again, I will not go into details at this point. We will discuss each one of these examples in greater detail at a later stage during this particular course. Now again, just as we have seen in the case of the glucocorticoid receptor, this NF kappa B is actually present in inactive form in the cytoplasm. The NF kappa B is normally present in inactive form in the cytoplasm of cells, but in response to certain stimulus. For example, there are many situations where there is an inflammatory response, there is inflammation. In such cases, there are a number of genes which need to be activated in response to this inflammation. So, how do these genes are activated in response to inflammation? So, what happens during this inflammation in response to those inflammatory signals? This is what happens. Normally the protein, the NF kappa B is actually a heterodimer. Remember, this is this is another important point that you have to note down. Many of the eukaryotic transcription factors bind to DNA as dimers, okay, not as monomers, they actually bind as dimers. We will discuss this more detail in the next class. In the case of previous case, for example, the glucocorticoid receptor, it is called as a homodimer, because two monomers of glucocorticoid receptor actually go and bind to the DNA. So, it is called as a homodimer. But in the case of NF kappa B, it is an example of a heterodimeric transcription factor, where it has two different subunits, one is called as P65, another is called as a P50. So, while glucocorticoid receptor is a homodimer, NF kappa B is a heterodimeric transcription factor. Now, but I want to give this example because you can see the mechanism of nuclear translation of glucocorticoid receptor is different. The interaction between HSP90 and glucocorticoid receptor is abolished when the hormone actually binds, and that is how the protein goes in the nucleus. But in this case, the protein is prevented from going to the nucleus because it is interacting with another protein called I kappa B. It is called I kappa B because it is called inhibitor of kappa B, okay, nuclear NF kappa B. Now, as long as this heterodimeric NF kappa B is interacting with the I kappa B, the protein stays in the cytoplasm. Now, when there are specific situations like an inflammatory response, when you have to activate the genes in response to this kind of inflammatory signals, now this nuclear NF kappa B has to be translocated inside the nucleus. Now, what happens? Through a cascade of events, which again we will discuss later, this I kappa B protein gets phosphorylated. Now, when the I kappa B gets phosphorylated, the phosphorylated form of I kappa B is actually the substrate for what is called as a ubiquitinylation of I kappa B. Again, we will discuss these things later. Ubiquitinylation is another important post translation modification, and usually proteins which are ubiquitinated, they are degraded. So, ubiquitinylation of protein is actually a signal for it to be taken to a degradation pathway. So, once the I kappa B is ubiquitinated, the protein degradation machinery goes and degrades the I kappa B, and therefore, in the absence of I kappa B, the nuclear factor actually the NF kappa B now can go inside the nucleus, bind to specific response elements called as NF kappa B response elements in of various promoters of genes which are actually need to be expressed in response to inflammation, and all these genes are getting activated. So, I have given all these various examples just to tell you that over the years, using the various experimental approaches I have described, people have started understanding how various eukaryotic genes are getting activated. So, I gave some 4 examples, but you can go and then find out a number of such examples. So, as I can show here, there are now families of transcription factors. There are a number of eukaryotic transcription factor families, and many times these transcription factor families have been named based on the kind of DNA binding domain they have. I told you the transcription factor contains a DNA binding domain and a transcription activator domain, and depending upon what kind of DNA binding domain they have, these various transcription factors have been classified into, for example, homeodomain family, basic helix loop helix family, basic leucine zipper family, zinc finger family, and so on and so forth. We will discuss these things more detail in the next class. So, what is the crux of the matter what I told you so far is that transcriptional activation in mammals depends on more than 2000 site specific transcription factors which interact with general transcription machinery either directly or through intermediary proteins known as coactivators, and as a result of this transcription activation actually takes place. Now, again I have not touched so far about the interaction of the transcription factor with general transcription machinery. How exactly the interaction takes place, we will discuss couple of classes later. Okay. So far I have been telling you about only binding to a specific DNA sequence, but once the transcription factor binds to a specific DNA sequence, it has to interact with the general transcription machinery and RNA polymerase. That itself is a huge problem which has been solved. And how exactly they interact? Do they directly interact with general transcription factors, or do they interact through certain proteins called as coactivators or co-recruiters? We will discuss much later. So, coming back to this point, as development and differentiation proceeds, selection among these more than 2000 transcription factors ensues to the regulation of cell specific gene expression. So, there are more than 2000 transcription factors, each of which are selected at different time points during development or during the adult stem, and these combination of these transcription factors then go and activate the transcription of specific genes. And this kind of a differential gene regulation is very important during embryonic development. Again we will discuss this later, much later. So, during embryonic development, promoters of specific genes contain transcription factor binding sites that are often organized into clusters called cis regulatory modules or CRMs. Now, so far I have been talking about only one particular element. I told you glucocorticoid receptor goes and binds to this element and activates transcription. Heat shock transcription factor goes and binds to heat shock element and activates transcription. But during development, if you look at certain promoters of certain genes, it is not one or two elements, there are combination of multiple cis acting regulatory elements to which a number of different sequence specific DNA binding proteins or sequence specific transcription factors bind, and it is the synergistic effect of all these transcription factors that ultimately leads to activation of that particular gene. Okay. So, developmental regulation of gene expression is much more complicated, which involves very complex cis acting regulatory elements called as cis regulatory modules or CRMs, which contain multiple transcription factor binding sites, and the synergistic action of all these things ultimately lead to transcription activation. Now, I am going to just take you 2 or 3 minutes to explain to you that, so people can ask, okay, you have studied all these promoters, you have identified so many transcription factors, has there been any benefit arising out of this? What kind of benefits have we got it? Okay. I will just give you one example. For example, from all that I have told you now, it is becoming very clear that we have what are called as promoters. When transcription factors go and bind to these promoters, the downstream gene gets activated. Now, this principle itself has now led to what is called as production of recombinant proteins in a expression system of your choice. So, understanding promoters and transcription factors has helped us to develop expression systems to produce a number of recombinant proteins. For example, you want to make insulin, you want to make growth hormone, you want to make recombinant hepatitis B vaccine by expressing hepatitis B antigen, or you want to make factor VIII, which is a very important clotting factor. All that what you have to do, you have to take the gene coding for these proteins and then put in front of promoter of your choice. For example, you want to make a protein in bacteria, you put a bacterial promoter and put this plasmid in bacterial cells, now bacteria will start making your protein of your interest. On the contrary, if you want to make this protein in yeast cells, you simply put an yeast promoter, now yeast cells will start making your protein of your interest. Similarly, if you put an insect promoter, your protein will be made in insect cells, or if you put a mammalian promoter, your protein can be made in mammalian cells, or if you put a plant promoter, you can make your protein of interest plant cells. And you can see, so this particular ability to make recombinant proteins in an expression system of your choice has now become a billion-dollar industry. No, recombinant protein production, growth hormone, hepatitis B antigen, insulin, various cytokines, there are so many useful proteins can now be made in expression systems in mammalian cells, plant cells, insect cells, bacterial cells, and can be used for diagnostic and therapeutic purposes. And if you actually go to this particular website, it actually tells you that the US Food and Drug Administration has actually approved more than 200 such recombinant proteins or peptides which have been produced using this recombinant DNA technology. Okay. So, in addition, there are many transcription factors and promoter elements themselves are actually targets for a number of diseases. By turning on a specific promoter or turning off a specific promoter, you can actually prevent or cure a particular disease. Again, we will discuss this in greater detail as we understand a little bit better about how transcription factors function. So, remember, there have been a number of benefits arising out of all these studies on eukaryotic gene expression. So, I think I will end my talk here. So, what I actually told you so far is that in addition to the core promoter elements and general transcription factors binding at the near upstream region, there are multiple upstream sequences called enhancers or proximal or distal promoter elements to which specific sequence specific transcription factors bind, and as a result of all these things, ultimately the rate of initiation of transcription of a particular eukaryotic gene is either upregulated or downregulated. Thank you.